Thanks again, everybody, for joining our seminar. Today we have a talk by Dr. Brandon Oubre, who is a research fellow at the MGH Department of Neurology and Harvard Medical School in the Laboratory for Deep Neurophenotyping, led by Dr. Anupam Gupta. Dr. Oubre received his PhD from the University of Massachusetts Amherst, Downing College of Computer and Information Sciences, with an outstanding dissertation award. And today he will be speaking to us about digital and quantitative behavioral phenotyping in neurological disease.
All right, thank you for the introduction. Anupam would love to be here today as well, but he always has a clinical conflict with the seminar series, so he sends his well wishes. So I have a mostly computational background, but for the past several years, I've been interested in this problem of how do we quantify human behavior specifically to pull out signs of disease that are present in that behavior. So really important to neurology, for instance. You could think of other disease areas where this is important, but just again, for neurology, this is a main focus. I think I have to click to make my clicker work. Yeah, so why is this such an interesting space to be in right now? There's a lot of reasons, but the accelerating pace of drug development is a key driver here. So, who here 20 years ago would have thought that we'd be hearing about, CRISPR-based gene therapies for sickle cell? It sounds like sci-fi, right? Getting more towards the neurospace. Again, after 20 years, not a lot of, no major breakthroughs for Alzheimer's. Now we're seeing potentially disease-modifying treatments. We're seeing progress in orphan diseases like Friedreich's ataxia. I think there was just a pill that did symptomatic relief. And then ASOs are also showing a lot of promise in the neurospace. There was Spinraza for SMA a while back, but there's still a lot of promise in potentially disease-modifying treatments for nucleotide repeat disorders using ASOs. So think Huntington's or ataxias. Lots of cool stuff here. Obviously, this all needs clinical trials. Clinical trials are long, they're expensive. We would like to make them more cost-effective so we can, run more studies. Also, and so many ways to get to that more cost-effective, but one way is to reduce the number of subjects in the trial. And in order to do that and still reach statistical significance, we need more sensitive measures. Another side effect of having smaller trials is we can start to run studies in some of these orphan diseases, right? Where maybe right now you can't even recruit enough subjects to run the study in the first place. So double whammy right there. And so when we're talking about more sensitive measures, what do we currently have to work? So in neurology, generally things are assessed using clinical motor assessments. So here's an example. A trained assessor, such as a neurologist, is going to ask you to perform a task. So here we have someone who's obviously not a control, drawing this Archimedes spiral. And they'll give you an ordinal score based on what they see. How your movements performed, or in this case, what you see in the image. And, they'll give you that score. You'll do a bunch of different tasks, maybe across different motor domains. You'll add up all those scores, and then that's your assessment score. There's obviously a lot of places where we can improve on this. For one, if we're talking about more sensitive measures, having 5 options is going to be a no-go, right? There's a lot of information here that even just by looking at this, you can start to pick out. And we're really collapsing it into just a few possibilities. Also, different centers, start to do these in different ways. For example, MGH likes to do these Archimedes spirals on the inside in the center. Some other centers like to do them on the actual line. And so you start to get these little variances across physicians, across different centers that make analyzing this data at scale hard. And then, finally, one thing that might not be immediately obvious is that these tasks are pretty contrived. They're made to elucidate a particular, cardinal sign of some disease, but they don't really reflect what's going on in the real world in your natural environment. And we really want to know, how does the disease impact your daily life? Because that's ultimately where we're trying to improve, right, is your daily life. And so, right now we have this different set of tools for trying to get at that question on patient-reported motor outcomes, PROMs. These are basically Likert-style surveys. You'll answer a couple of questions about how easy it is to do a certain task, or just specifically, how is this disease, impacting this, right? And the nice thing about these is they're more scalable than the clinical motor scales, but you can only do them so frequently, right? If you try to do these, every 30 minutes, for example, someone's going to get really annoyed at you, or just, even daily, they'll just fill out, the same answers without really thinking about it. So an important tool, but obviously we want this to be more objective, because it's a subjective thing. And when you start to look at, for example, populations with cognitive issues or memory loss, are they really answering these accurately? Or even comparing the more severe subjects to the ones that are less severe, maybe a caregiver has to fill out the form. And so there's all of this subjectivity and things going on with all of these forms that we'd like to get away from. Technology obviously gives us a way to try to address a lot of these issues. And when we think digital measures, you can think both a signal from some sensing system. There's a whole bunch of different options, create a whole bunch of different ideas, but then also the algorithms and processes that are needed to turn that signal into something clinically meaningful. And so just to give you a brief grounding that there really is some signal to capture, here's a paper from a couple years ago where patients were asked to do this saccadic eye movements task. So just looking at a dot on the screen that moves from side to side. And their gaze was captured using a smartphone camera, just commercial off-the-shelf software. And you can see here clearly at the bottom is somebody with more or less normal saccades. And at the top is somebody with these very inaccurate saccades. And so there's clearly signal here, even at a very high level overview, that we can take advantage of. And so there's two broad potential ways that we can try to improve clinical measures. There's the sensitivity, like I talked about. There's also this question of ecological validity, which is what the proms are getting into. But for now, I think let's just focus on the sensitivity. And I want to talk a bit about part of what we're doing at MGH right now to address this issue of how do we create more sensitive measures. And part of the way we're doing that is the Neurobooth. It's this really cool system. You could Google this and see some images and a bunch of stuff, but it's a telephone-sized booth that's actually sitting in the clinic, across the street at MGH. And we have all kinds of different sensors in there. We have, high-end sensors. We have commercial sensors. We have audio, video, inertial data, eye tracking, and all of this is time synchronized. So just a really cool data set. You don't usually in the literature, you only really see like one of these things, right? Not all of them together. But then also the really nice thing about this is its scalability. So, patients come in for 30 minutes before or after their appointment, and that means we can really collect a lot of data. And data is really what's holding back a lot of this research. It's privacy constrained. And so just in the last, year, we've had, several hundred unique participants. We're getting in a longitudinal of 3, 4, 5 sessions. So really cool data set. I want to quickly acknowledge this is a very large effort. It's not just a couple people. Anupum's running the whole project, but we have a bunch of software developers and postdocs developing the software. Making sure things are running well, analyzing the data. Clinical research coordinators actually collecting the data. Clinical collaborators, engineering collaborators, all helping to make this thing work. But yeah, there's a lot of data, really cool to look at. But just to show you some of what we're seeing, I want to zoom in a little bit to just the ocular motor time series, that this eye tracking. Why eye tracking? One is that you see ocular motor deficits really early on in a lot of diseases. And so, it gives us that, ability for early disease detection. Also, eye movements are very small and fast. And so you can see there's a big gap between what we as humans can perceive in the clinical motor scales and what the technology is able to capture. So there's a lot of room for improvement over the existing state of the art with this particular stream. And then finally, there's decades of literature on eye movements to rely upon. But with this, very large longitudinal set in a lot of, particularly rare disease populations, there's still a lot of low-hanging fruit that we can really get at. And so on the right here, I'm just showing you some of this clear signal where, we have the controls on the leftmost, time series column. And on the right, we see just very clear signs of disease. These time series look very different. Of course, I've cherry-picked some of the more severe ones. But you can see really interesting signals. And so, this gets into this question of, computationally, what's the best way to model these? And I really want to emphasize, in this data set, even though we have a lot of data, we're still constrained in a lot of ways, right? We can't just take the latest machine learning model, from the latest paper and throw it at the data. because we don't have the whole internet to work with, right? We have relatively precious few data points, even with this hundreds of subjects, right? To try to learn these features. And so, if you have to think about, how do I bake the inductive biases into the model, and where do they make most sense? So just one example of the level at which we tend to think about these things. Let's say you have a network for temporal modeling. Generally, your structure is going to be something like, let's segment the time series. For each segment, let's have some lower layers of our network that encode some latent vector, some structure, right? And then let's have the higher levels try to model temporal context across these layers. But even at that basic question of let's segment the window, segment the data, there's a lot of options, right? Most of your ML approaches are going to do some sort of fixed length sliding windows. But with eye movements, we know eye movements for many tasks are basically grouped into saccades and fixations. So you could think about using that instead to reflect what we know about how they move. And just some trade-offs here, you can think about are, with the sliding windows, there's less of an inductive bias, so you could potentially learn new phenomena, but you're going to need more data to do that, right? Just one example of why you might need more data is, thinking about what input those very early layers in the network get, right? With just a fixed length sliding window, you're going to have a lot of variability compared to, oh, this is a saccade. We know saccades have certain shapes, right? This is a fixation. We know something about what a fixation looks like. So that more variability, again, means more data. But then also, even when you get into very simple questions like, let's just use the sliding window, but what should the size be, right? That can be deceptively hard to fine tune for some of these data sets because, obviously the size is too small. You're not giving those initial layers enough to learn from. It's too big. You're trying to do too much temporal modeling too early on in your network, right? You're not developing good representations because of that data variability. But then also just exactly how big you make the window is going to influence what kind of features you can pull out of the data, right? What level of, let's say you have really fast disease signal and a somewhat slower disease signal. How do you optimize picking out both of those, right? Whereas on the flip side, if we take something based on our knowledge of eye movements, we do this at cause of fixations. Now you're introducing this additional pre-processing step. You have to really trust that saccade detection algorithm and understand its quirks, right? So again, just you need to really understand the data and try to figure out, what's the best way. So one way that we kind of tried to approach this was let's take, the approach with the less inductive bias and just do a simple model and see what it learns, right? So here, what I'm doing is a very simple network where we're analyzing each window of the data independently with just fixed length sliding windows. And we're predicting whether or not we think this window comes from somebody with control, who's a control or has this disease. I'm not going to explain it yet, but just ataxia, right? And we just want to see, this is very simple model with no long-term temporal modeling. Is that able to do something interesting? And what's nice about this is, you can just plot the time series. So on the top here, I have the position of the eye. And on the bottom, I have the velocity. The model's only seeing the velocity here. And then the color just corresponds to, what the model thinks about each window. And so you can see here, we're getting some cooler colors for, what is basically this really cardinal disease sign called gaze of nystagmus. You see the sawtooth pattern, right? So really cool that the model's picking up that with really, not a lot of information that we're giving it. And also you see here, we have this very inaccurate saccade that the model's picking up on. So it's learning something. You could try to just aggregate these very simply without some, complicated methodology. So, you could take the mean, but obviously there's a lot of 0.5s here. It's not a great idea. Another approach is you could just try to learn, learn a summary score based on the distribution of the probabilities. If you do that, what you start to see is, you look at the model probabilities with respect to the severity of the I-score. So, controls and then increasing severity as the score gets larger. And you see that the model probabilities tend to increase as the severity gets worse, even though we didn't explicitly tell the model anything about the severity. And so this model's learning something. That's cool. We actually see some signal. But if you start to look at this closer, you'll see that there's actually this, cooler regions that correspond to seemingly normal saccades. And if you start to look at some of these tasks more broadly, you'll notice for a lot of these fixational tasks, it's developing a saccade detector. For these smooth pursuit tasks, it looks like the model's developing a saccade detector. And these make sense because in those tasks, the saccades are actually going to be indicative of disease. You're supposed to be having a smooth pursuit, or you're supposed to be fixing relatively still. And so it makes sense that you'd be doing this. And so then maybe you say, okay, here's the data evidence that really, the model's developing the saccade detector anyway. We should start to go with the second option of let's model based on the saccade fixations. If you do that, it's you want to look at data that has a lot of saccades in the data. And so here's just an example of a passage reading task where somebody is reading this fixed passage on the screen. And as they do that, we're tracking their eye movements. We're also tracking their speech. They're being asked to say this out loud. So in the future, there's this great opportunity for, looking at the synchronization of these time series. But also, this is a more naturalistic task and less contrived. So really interesting to look at. And so just to orient you, what we're seeing here in the gaze data is the staircase pattern, where you make saccades left to right on the screen as you read the line. Every now and then you make a regression. You go back and look at a previous word. This happens in controls, but it happens more frequently if you have trouble understanding the word. So these regressions are usually triggered by cognitive and lexical processing processes. Eventually, you get to the end of the line, you do a return sweep, and then you start looking at the next line. So yeah, that's what the data looks like. And then let's just look very at a high level at the data. Let's just look to see if there is any low level temporal pattern that is informative. And so what we're looking at here are basic triplets of gaze events. So just looking at a saccade followed by a fixation and then a saccade, right? So when you're looking at a word and when you're transitioning between words, just as triplets. And so what's really cool here is if you just look at a t-SNE, you see the structure of the task pop out. So that's giving you some confidence that the saccade detection's working well. And also that, you can start to see some characteristics of the task. So just you see some subclusters here, for example, where these really large saccades happen, these are your return sweeps. And so the return sweeps are being very, very accurately captured in the data. They're being set out apart. So we're learning something cool there, plugging the structure. Also, just, if we look at the duration and the displacement of those fixations, you can start to see that, high duration fixations and high displacement fixations kind of start to get grouped together, even though they're not overlapping. One thing I want to point out is this, what is a high displacement fixation? Your eye is actually doing a lot of things while it's supposed to be, reading. It fixed at one point. There's actually some drift, some small scale nystagmus. But then as part of, larger eye movements, basically if it's not a saccade, it's a fixation. So there's a bit of a terminology gap here, right? But then also one cool thing we're seeing here is this little region out here with these very high displacements. This is actually saying, there's an error in the saccade detection algorithm. These are all saccades that got mislabeled as fixations. So just one cool thing popping out of there, you can see that those are kind of being quarantined to some extent, right? But then what was really interesting is that just based on these basic kinematic features, you actually start to see where some of these high displacement fixations happen, we actually see relationship to disease severity. So I've plotted the bars, this clinical motor scale, ocular motor score, in reverse order to where controls show up on top. And you start to see that we, in some of these regions, we don't see any control data or low-level disease data in these spots. And so we can also just look at the probability of a particular region being from somebody with ataxia versus somebody who's a control. And you start to see, these regions correspond to more likely to be somebody with this disease. And so there's obviously information, related to severity just based on simple kinematic features. So then, you can go ahead and just train a simple, random forest just to make sure, what we're seeing is real. There is a relationship here between disease information and basics, triplet kinematics. This is very low level understanding the data. How do we bake in the temporal model? But hopefully it's clear that there is information here to learn. And then it starts to open up these more interesting questions. First of all, how do we actually model the full time series? What a transformer work here, for example, right? What's the right way of doing this? But also, side questions of, can we actually learn the structure of the task from normative data? Because there's a lot of potential data online on different data sets of people just reading. Can we use that to understand the structure of the task and then make more efficient use of our harder to get disease data, right? There's some caveats and pitfalls there, but that would be really nice if we could. But then also, just taking a page out of more modern machine learning techniques where we pre-train a model, then fine-tune it to a particular subtask. Can we learn something about a common representation that we can fine-tune to say, okay, let's just look at early disease. Can we differentiate pre-symptomatic from controls? Or let's look at a larger picture in the feature space. We want to figure out severity, right? And so basically, yeah, what are the best ways of learning these things, and what are the ways that we can start to answer this question without just our basic supervised regression throwing in the clinical scores? Because there are some problems with that I'll talk about later. Yeah, so that's just some of the data that we have towards that idea of more sensitive progression. Taking a step back, this idea of ecologically valid measures, right? How do I naturally interact with technology, and what can that tell me about disease science? Is another interesting question that we want to answer. So, when you think about technology in home settings, you have to think about the burdens that it imposes on the people using it. So one is just the burden of the instrumentation, right? If you're going out wearing a million different wearable sensors, maybe no one wants to do that, right? It's uncomfortable. Maybe you don't like the camera in your living room recording your, right? A privacy concern would fall into this. Then also, a lot of research, or earlier research at least, is focusing more on the frequent measurement, but asking people to perform these subsets of clinical motor scales. Whereas what we really want, again, is to measure natural interactions with technology. We don't want somebody to have to go out of their way to perform a particular task. We want to seamlessly capture what's already happening. So there's this continuum of, we know the context of what's going on, and we can leverage that versus we're just kind of looking at everything. But then, how that context is important. And so how do you really get to this idea of looking at everything? And one of the key insights here is that movements actually have a lot of regularity to them. You can visually discern, healthy control versus somebody with a disease, and we rely on that for the existing clinical motor scales. And so, why is that? Part of it is because the nervous system is doing a lot of optimization. You're optimizing for smoothness, you're optimizing for energy expenditure, right? And so you get these regular patterns. And in motor control, there's this theory that you're actually taking these building blocks, these sub movements, and stitching those together to create more complex, general movements. And the idea here is, if there is consistent patterns to those sub movements, consistent kinematics and healthy controls, consistent shapes of the velocity space, right? Then if there's deviations from those healthy patterns, we can start to measure those to get at disease, right? And so the shift here is, instead of analyzing a particular movement, we analyze the components of a movement that should form any type of movement, right? So just a shift in what we consider as part of our analysis unit. And so the basic question, right off the bat is, is there actually enough information here to estimate severity? And we're looking at ataxia in this particular case. And so just for reference, ataxia is a neurologic phenotype that arises when you have an impairment to the cerebellum. So, the cerebellum's, back of your head region right here. And on screen right here, we have somebody with a normal cerebellum. And then on the left, we have somebody with a neurodegenerative disease that is obviously atrophying, right? And so part of what is nice about the ataxia space from an analysis perspective is that the phenotype that we see in movement is focal to the cerebellum. with other diseases like stroke, you might be looking at different signs depending on exactly where in the brain the lesion happened. But then also one of the cardinal signs of ataxia is this decomposition of movement, which just happens to fit in really well with this idea of looking at the building blocks. And so just starting off to answer the question, is there information here? We asked people to perform this finger nose task between your nose and a tablet. This is pretty closely aligned to what clinicians will actually do in a clinical setting as part of these clinical motor scores. But also it's this, nice upper limb task. And so there's somewhat of a parallel between this and the kinds of motions you might, use to interact with your environment in a naturalistic setting. And so, yeah, just looking at the kinematic features of these submovements, you start to see some really clear trends. So what I'm showing you here, each column, the first column is submovement duration. The middle one is submovement speed. The right one is submovement displacements. And then on the top, we have aggregations for each subject. And on the bottom, we just pool the individual movement elements so you can see the density estimates. And just on the top, you see very clear trends, reducing duration, reducing speed, reducing distance as a function of severity. Then also what's really nice here is this peak in this histogram shows very nicely the region that's corresponding to these large back and forth movements of the task. And as the disease increases, that breaks down. We see a lot less density there, and it shifts towards smaller, more decomposed movements, which is exactly what we expect to see with the disease. So just very nice, clear kinematic trends, also trends related to the shapes of these things and the temporal sequence of these things. But just for time's sake, those are the easiest ones to look at. So then what we can do here is we can just train our regression model. Say, let's take the clinician score as our labels, and on the y-axis, the model output as our estimates, right? And we do a decent job here. I think the takeaway isn't like, okay, yeah, you estimated the scale based on maybe just one component of it, right? That is an interesting finding, but I think the real way of looking at this is we're showing convergent validity between the information in these movement elements, these submovements, and what we know as a proxy of disease severity, the clinical score, right? And so these are capturing similar things, which is really the thing we were seeking to answer. Also here, we happen to have some longitudinal data. One of the drawbacks to a lot of this research is, it's very cross-sectional because it's hard to collect the data. But here we had a few longitudinal data points. And so what we're looking at is the model's estimate from the nth visit compared to the nth plus 1th visit. And you see there's a decent bit of agreement here. What's also interesting is when there's a divergence, it happens to be more in the direction of disease progression. And so this isn't really enough data to claim like, hey, the model's capturing progression, even though the clinical score isn't. But it is in that direction. So it's at least a promising sign. But overall, the model estimates are fairly consistent, even though it was trained in a completely cross-sectional manner. So in summary, we can estimate this overall severity, but we're doing it from this, large continuous movement. So how can we get away from that? How can we get away from these large movements that don't really reflect what people will do in a real-world setting? So then there's this other study that the lab did, basically instrumenting an accelerometer on the wrist and on the ankle. People went about their daily life for, I think it was 6 to 7 days. And then you can see some of the cool data coming out of that. Obviously, people sleep. So, you need to do some additional pre-processing, pull out sleep, just look at, daily living. People also sit on the couch and do nothing. So you need to, do some processing to figure out when are people actually moving. So you can define some activity bouts. And then during these periods of activity, you can start to pull out these submovements, the same types of things that we're looking at in this previous work. And then we start to see you do these submovements from this naturalistic data start to reflect disease severity. And they do. I'll go to some additional plots here. On the left here, we have two different scales for measuring disease severity, clinical motor scales. And again, we see correlation between the regression model outputs and the severity of the disease. But then also we see correlation between the model outputs and these patient-reported motor outcomes. And so we're actually seeing agreement between what happens in the home and what the clinician sees. And so we're measuring similar concepts to both of these. Again, this idea of convergent validity. But then also, if you train the model to say, let's take the first 3 days and compare the model estimate to the second 3 days, we get really nice agreement between the two outputs. And so, this model is saying consistent things, based on a day-to-day basis. And then, of course, we can also just compare, what are the model say for controls versus what does it say for the disease group. And we see a clear difference. We're not severely overestimating controls. So cool. We see evidence of disease severity in this free living data. We're doing it in an ecologically valid manner. People are just, going about their day. But then what about disease progression? The key concept here is that the features that we see that are most related to severity aren't necessarily going to be the ones that are most indicative about progression of the disease. So, part of this is we need the longitudinal data to actually pull this out. And so that's where we took a step away from the ataxia and started looking at ALS. So ALS TDI has this, really great, really nice large data set of people with ALS, basically wearing sensors on the wrists and ankles, just again, daily life. So just to give you an idea of some of the scale here, vertically we have each subject, and then horizontally we have each data collection. And there's the obvious COVID gap here. Then so for each one of these sessions, you have a lot of data, days worth of data potentially. You could do the same kind of modeling, right? You can get rid of sleep, you can find periods of activity, extract these sub-movements, and then you can either try to do basic regression like we did before, or you can start to think, how do I model these in a different way that tries to capture progression, right? And so the way that Anna Poon went about this was, let's look at time points from within one subject's trajectory, and then make the assumption, because this is a neurodegenerative disease, that time point 2 is going to be more severe than time point 1, right? Later time points are going to be more severe because the disease has had time to progress. And then you can basically subtract the feature vectors and to get the direction and feature space of progression, right? And then label it as, was this a prior instance or a subsequent instance? And if you do that, you actually start to see some really cool trends. So, here on the top, we have the estimates from these pairwise models. And on the bottom, we have the clinical scores. And you can see that at least visually for these instances, that the pairwise models seem to be picking up evidence of progression, even though we're not seeing quite that much change in the clinician models. And then also for this particular case, you're seeing a lot of agreement, similar rates of progression between the wrists and the ankles. But then if you look at some other subjects, you start to see a divergence where their gait, for example, their ankles are actually progressing at a worse rate or a slower rate than what we see in their upper limb movements captured by the wrist. I mean, again, even though we're seeing this nice, differential pattern here, it's not necessarily captured by the clinical motor scores. Just pulling back to a full high-level overview, you can do the stats, and it shows that these models, based on these wearable sensors, are actually able to capture the rate of progression more sensitively than the existing clinical scales. And again, we're doing this in an ecologically valid way where we're capturing natural behavior as opposed to a more contrived behavior. So yeah, overall, very cool result. It's not the end of the story. There's still plenty of things you can do. For example, we're just looking at inertial data here, but what about speech? What about eye movements, right? I said earlier that, eye movements can start to show signs of disease earlier than some of these other domains, right? So how do we, integrate the data from all these different motor domains like a clinical scale does and start to get an even more sensitive idea of progression, right? So just one way in which you could take this, Also zooming back a little bit, these pairwise models are actually fairly simple from a modeling machine learning perspective. If you sit down and think about these for a while, there's a lot of potential ways where you can try to model this in a more sophisticated manner, potentially get even better measures. So yeah, kind of wrapping things up. What are some ways to move forward overall? Where are some key ideal spots for collaboration with ML for health communities, right? First is this idea of how do we actually learn and what do we want to learn, right? A lot of the research in this space functions by trying to learn based on the clinical scores as your supervised target labels. That we don't have this problem for the pairwise regressions I just talked about, but it is a consistent problem. And why this is a problem is, let's say I were to give you a plot that says, here's your training labels, here's your model estimates. They have perfect agreement. Is that good? In your general case, yeah, great. You learned the function perfectly well, right? But in the case of trying to develop better measures, that's actually a problem, because if you get perfect agreement to your labels, all you've really done is replicate all the flaws of the scale you're trying to improve. So how do we get out of that paradigm, right? Also, just, I showed you earlier that those scores are really coarse. And so if you use those as your training labels and you're trying to get something more sensitive, all you're really doing is introducing noise into your learning process, right? So the pairwise models is one way to get around that. But going back to thinking more broadly, how do we model the data in a way that doesn't have these fundamental limitations? Just kind of the easy supervised regression to get these more sensitive measures. Also, I know ML for Health and the Broad community does a lot of imaging and genetics, and there's this, I think, adding in this component of the phenotype that that we see through these sensors is an important part of how we can start to understand the disease state, right? Do we start to see differences in these phenotypes based on genotype, or can we use the phenotypic differences to try to identify genes, right? If we're talking about more sensitive measures, we have all of this wearable data, for instance, but does the imaging or does what we know about the genetics give us an even better idea of the sensitivity, right? If we know somebody has, say, a disease a gene that modifies the disease progression, we can have a separate model for them, right? We can model them more accurately. So yeah, a lot of potential overlap in how do we take this disparate data that's maybe reminiscent of multi-omics doing it, right? And then finally, just more broadly, we're trying to create some of these more ecologically valid, more sensitive measures for use in clinical trials. If there's any, if out of the genetics or out of the imaging comes some potential intervention, I think there's a lot of potential for using this kind of technology to assess, does whatever intervention actually work in improving disease state, especially if you start to get into things like N of 1 trials for ultra rare diseases, right? How do you know this custom treatment is working for this one person, kind of a deal, right? I think there's a lot of potential for collaboration there. Yeah. Just finally, I wanted to thank a lot of people. Anna Poom, Ivan, and John Francois were mentors to me, and their contributions were to a lot of the work here. Also, I had their pictures earlier on the slides, but Sid, Anna, and Nicole led a lot of the studies on this ecologically valid monitoring track, as well as we used a lot of the data from the ALS TDI. Yeah, I think I ended early. Thank you.